Now today we are going to discuss a very important topic again from examination point of view and a very interesting topic. Today we will be starting up with vitamins. Now being dental students, you should know what is the value of vitamins, what role these vitamins play in our body. Right. So vitamins are actually, if we look at the definition, we can say that vitamins are the compounds which are required for the normal functioning of the body. And broadly, this vitamin term was given by scientist Hunk. And these vitamins are categorized into two types of classes. One class is fat soluble vitamins. And second is water soluble vitamins. Now, as it is clear from the word itself, fat soluble means which are soluble in fats. And what are fats? Fats are the again organic compounds which are insoluble in water. And that is why if we look at fat soluble vitamins, their absorption is different. Their storage is different, and obviously their transportation will be different just because of one simple reason, that they are insoluble in water and they are soluble in organic solvents. So whenever they come into circulation, they will always seek for a special protein which can transport them. Coming over to water soluble, it is clear from the word itself, which are soluble in water. Right. So since they are soluble, they are not stored as such except B12. Otherwise, once their function is over, they are excreted in the urine. Okay. So if we look at the type of fat soluble and water soluble vitamins, fat soluble are A, D, E and K. And always remember, you have to focus on the active form of the vitamin. This is how the questions are asked. You should know what is the active form of a particular vitamin, what are the biochemical functions, and what are the deficiency manifestations if a particular vitamin is absent in the diet. So if you look at vitamin A, it has three active forms, but the most important one is it is retinol. If we look at vitamin D, the active form is calcitriol. If we look at vitamin E, active form is tocopherol. And vitamin K is phylloquinone. And we will be discussing in detail also this vitamin K has further three forms. And one is a water soluble form also. Coming over to water soluble vitamins, always remember water soluble means vitamin B complex and vitamin C. They are water soluble vitamins. Right. Now, if we look at B complex, it consists of, again, we can differentiate it on the basis of two classes again, hematopoietic and non-hematopoietic. Right. And what are B complex vitamins? If we talk about hematopoietic, B9, which is also known as folic acid, and B12, which is also known as cobalamin. And if we talk about non-hematopoietic, you start from here, B1, whose active form is TPP and it is also known as thiamine. Then comes B2, which is also known as riboflavin. B3, which is known as niacin. B5, which is known as pentothenate, and B6 is known as pyridoxine. B7 is known as biotin. Right? B9 and B12 we have already discussed, and comes C, which is also known as ascorbic acid. Right? Now, what are the important points which you people should remember about fat-soluble and water-soluble vitamins? Now, see, the important features are, always remember that if we talk about water-soluble, all water-soluble vitamins functions as coenzymes. Coenzymes, and we have already discussed what are coenzymes, which work in cooperation with the enzyme. And you will see that all the vitamins which belong to B complex category, they will be used as most important coenzymes. Whereas in fat-soluble vitamins, only vitamin K functions as a coenzyme. Always remember this. This is one of the very, very important differences between water-soluble and fat-soluble. Okay? In fat-soluble, only K will function as a coenzyme, whereas you will see when we will discuss the functions of all water-soluble vitamins, they are going to function as coenzymes. Now, there is a very interesting chart of water-soluble and fat-soluble vitamins. Now, see, if we look at, this is a very interesting one, if we look at the solubility, you can see that fat-soluble are fat-soluble, obviously soluble in fat, water are in water. If we talk about absorption, I told you that absorption means since they are not soluble in water, they will always require a carrier protein. So absorption ke liye obviously bile salts, because bile salts are the ones which are required for the emulsification of fats. So you can say that absorption mein bile salts are required, whereas in water soluble simple intestinal absorption is there. Coming over to transportation, since they are not soluble, they require always a carrier protein to be transported. So the difference is transported by a carrier protein, and here it travels freely in the body without requiring carrier protein, except obviously vitamin B12, because I told you vitamin B12 is also stored in the body, right? An intrinsic factor is required for its absorption and transportation. Then if we look at the storage, always remember, that is what I told you, fat-soluble vitamins are stored in the liver and the fatty tissues. And if we talk about water-soluble, only B12 is the one which is stored, rest are excreted in urine. Then if we talk about excretion, because they are stored, whereas if we talk about water soluble, they will be easily excreted in urine because they are soluble. Then comes accumulation. Accumulation is very important in a sense that there is always a recommended dietary allowance for every vitamin. Means how much dosage of vitamin is allowed in a day. So if it increases, it leads to hypervitaminosis. If it decreases, it leads to hypovitaminosis. When we talk about hypervitaminosis, it is very dangerous in fat soluble because they are stored. Right, we will be discussing hypervitaminosis of what clinical manifestations are there. That all we are going to discuss in this. Then if we talk about water soluble, you can say usually hypervitaminosis does not occur. And then except in high dosage and slow release of some B vitamins, but in general it does not occur. And the last difference is deficiency compensation. Deficiency compensation required in periodic doses, whereas it is required in frequent doses. The reason is because the B complex vitamins are functioning, a majority of them are functioning as the coenzymes. So this is a very interesting table between water soluble and fat soluble vitamins. Then coming over to, we are going to start up first of all with water soluble vitamins. Right? Now see, just now I have given you the entire list of water soluble vitamins. So first is vitamin B1. Now what you have to remember in this? First of all, you should know the active form of this. And then since I told you that they are functioning as coenzymes, so you should know that what are the reactions in which they are functioning as coenzymes. Because if they are absent, that reaction will suffer and it will lead to a particular disease. Clear? So vitamin B1, the active form, it is also known as thiamine, and don't confuse it with thiamine. Thiamine is B1, whereas thymine, just write thymine is the base of DNA. So the spelling is different. It is thiamine and pronunciation is also different. Now coming over to the active form of thiamine, active form is TPP, which stands for thiamine pyrophosphate. Or commonly it is known as TPP. Then comes the active sources, active or the rich sources. Rice unpolished. And the most of the sources of vitamin B complex are milk is there, then you can say butter is there, fish is there. These are the common sources. Green leafy vegetables is there, but this is the richest source. Then come over the biochemical functions. Now just now I told you that if we look at the biochemical functions, they are used as coenzymes. Right? Now what are the major reactions in which TPP functions as a coenzyme? So the first reaction is conversion of pyruvic acid to acetyl coenzyme A. A very important reaction, one-step reaction is there, means pyruvic acid, which has 3 carbon atoms, it gives out 1 molecule of carbon to form acetyl coenzyme A, which has 2 carbon atoms. So wherever you can say 1 molecule of carbon dioxide is lost, it is decarboxylation. And if I write the reaction is this. COOH, this is pyruvic acid, and it will form CH3, C double bond O, SCOA. The name of the enzyme here is PDH. This PDH stands for pyruvate dehydrogenase complex, right? And this is an example of multi-enzyme complex. Now, why it is known as an multi-enzyme complex? It is because it requires 5 major vitamin B only, which are B1, B2, B3, B5 and lipoic acid. These 5 are the major components of PDH. And that is why we refer them also as multivitamins. So whenever we talk about multivitamins, we are talking about this reaction. And the most important point about this reaction is B1, which is TPP, most important for cellular respiration. Because you know ये reaction कौन सा reaction है? This is a link reaction. किसके बीच में link reaction है? Reaction is linked between glycolysis and PCA. And you know that intermediary metabolism, TCA, is considered to be the most important pathway. It is known as an amphibolic pathway and it gives energy to the brain. So you can very well imagine the significance of TTP and how it is reacting with this. Clear? So this is a very important feature of, you can say, thymine pyrophosphate. Okay? And along with these 5 vitamins, you can say it requires 3 enzymes also. That is why it is an example of multi-enzyme complex. This is a very important question which has been asked in your exam so many times, either in this way or in some other way. Clear? So this is one reaction, conversion of pyruvic acid to acetyl coenzyme A. Second reaction in which it acts, and this process is oxidative decarboxylation. Now coming over to the second step, it is a component of alpha keto glutarate dehydrogenase. It is a component of alpha ketoglutarate dehydrogenase. This enzyme converts alpha keto glutarate to succinyl coenzyme A. This is also an enzyme of, again, a multi-enzyme complex. And if we look at the composition, the most important one is this TPP. And here also carbon dioxide is given out. Since this reaction is of TCA cycle, and since carbon dioxide is given out, it is also known as oxidative decarboxylation. So this is a second important reaction in which TPP is acting as a coenzyme. Then moving on to the third reaction, component of transketolase. Now, transketolase is a very, very important enzyme of HMP shunt. And this HMP shunt plays a very important role in maintaining integrity of RBC's membrane. And transketolase, simply you can say, if you remember, 5 carbon atoms plus 5 carbon atoms, they will split up into 7 carbon atom and 3 carbon atoms, right? And this 3 carbon atom, these two will again combine to split up into 6 carbon atoms and 4 carbon atoms, right? And this 4 carbon atom will again combine with 5 carbon atom to form 6 carbon atom and 3 carbon compound. So here 2 transketolase reactions are there and 1 transaldolase reaction is there. But whenever we talk about transketolase, transketolase will always require TPP. So this is third very important reaction catalyzed by TPP, again as a coenzyme. And last reaction, reaction number 4, is decarboxylation of branched chain amino acids. BCAA, branched chain amino acids. Again, the mnemonic of these branched chain amino acids is LIV, live. Leucine, isoleucine, and valine. All of them undergo 2 similar reactions. After that, their fate is decided because leucine is a typical ketogenic amino acid. Isoleucine is acting as both gluco as well as keto, whereas valine is a glucogenic amino acid. But before undergoing this fate, they have to undergo decarboxylation. Transamination, decarboxylation, and dehydrogenation. Decarboxylation के लिए TPP is the one which is required. So you can say that there are 4 important reactions in the body which cannot occur without TPP. First one is, let's go quickly see. First one is conversion of pyruvic acid to acetyl coenzyme A. The enzyme is PDH. The most important coenzyme is B1. Right? Because why it is, this question might strike your mind कि I have written B2, B3, B5 lipoic acid also. Why B1 is considered to be the important one? Why it is important? Because this is the one which takes out this carbon dioxide. Carbon dioxide is taken out by this. Rest reactions are like dehydrogenation and all, which are made possible by B2, B3, B5. Then second reaction is component of alpha ketoglutarate dehydrogenase, which is again a very important reaction of TCA cycle. You can see the reaction. Third is component of transketolase, which is a very important enzyme of HMP shunt. And then decarboxylation of BCAA. Again, the coenzyme is TPP and the enzyme is decarboxylase. So these are the four major reactions in our body which requires TPP. TPP for PDH, alpha ketoglutarate dehydrogenase, transketolase, and fourth one is BCAA. Clear? Now, these are the biochemical functions. Then after biochemical functions comes the deficiency manifestations. Now, if we look at the deficiency manifestations of TPP, the most important one is beriberi. Right? Now, this beriberi is of three types. One is wet beriberi. As it is clear from the word itself, wet. So wet से क्या होता है? Here edema will be there. Edema present. Right? And second is other neurological manifestations will be also there. So these are the features of wet beriberi. If we talk about dry beriberi, in dry beriberi, edema absent. So this is the main difference between wet and dry beriberi. But the other features will be like neurological manifestations will be there. Tachycardia will be there. The main reason is because these four reactions, the four you can say in which it is acting as a coenzyme, they are hampered, right? And then third is infantile beriberi. Now, what are the symptoms for infantile beriberi? Clinical features. I have again made a very interesting mnemonic. See. The mnemonic is berries. Berries, all of you eat, you know, the picture of berries is also given. It is B, breathlessness is there, big heart, dilated heart. E stands for eatlessness, which leads to anorexia. R, restlessness is also there. I for berries, IS, insomnia and soundlessness. So these are berries is the mnemonic and you can remember infantile beriberi. Now infantile beriberi kyun? If the mother is also deficient of B1, she is most likely to transfer this to the fetus. So you can say berries, breathlessness, big heart from B, eatlessness and restlessness, then insomnia and soundlessness. Okay? So this is what is the deficiency manifestations of vitamin B1 whose active form is TPP. Right? I'll just write here the mnemonic is berries. Now after this, another generally what we have seen is alcoholics are deficient of B1. The reason is that metabolism of alcohol interferes with it. Fares with B1 absorption. That is why they are deficient of this. Now see, what happens is कि if alcoholism is there and he is B1 deficient also, so it leads to Wernicke-Korsakoff syndrome. If a person is so much into alcohol and the person is suffering from B1 deficiency, what will happen is Wernicke-Korsakoff syndrome will be there. Whenever we talk about Wernicke's encephalopathy is there. Encephalopathy. Right? Nystagmus is there. And if we talk about Korsakoff, psychosis is there. Now, another important feature, what happens is कि if, and one very important thing is कि it can also lead to lactic acidosis. So if a person is alcoholic, right, and if he goes in hypoglycemia and he visits you, so what happens is the instantaneous reaction, instead of giving glucose, is thiamine supplementation first. I'll just write it down. Ki the person is already hypoglycemic because the concentration of glucose decreases, and the person is already high with alcohol. And if he is admitted or if he comes and visits you, so what happens is immediately what we have to do is we have to give thiamine supplementation. The reason is because this pyruvic acid is being converted into acetyl coenzyme A and it is forming lactic acid. Okay? This enzyme, because B1 alcohol is interfering with B1, so ये reaction possible नहीं है. So there is increased concentration of lactic acid. So immediately if that person is given this B1 supplementation, this reaction can be retained. And if this reaction is retained, what will happen? If this reaction is retained, acetyl coenzyme A will be produced, and this acetyl coenzyme A can undergo TCA, giving energy to the brain, and that alcoholic patient will start working. And obviously you can say gluconeogenesis will be there, glucose will be given instead of the formation of this lactic acid. Right? And this was the question which was asked in the exam also. So always remember this, because pyruvic acid to be converted into acetyl coenzyme A requires B1 as the most important coenzyme. Clear? So these are what? These are the deficiency manifestations of TPP. Beriberi, which can be wet. Wet may you have to remember edema is there. In dry beriberi, no edema is there. In fertile beriberi may always remember this mnemonic berries which I have discussed, and always remember alcoholics if they are deficient of B1, because B1 always both interfere with the absorption, so it predominates. Because alcohol metabolism में this NADH is required. And the enzyme here is aldehyde dehydrogenase or alcohol dehydrogenase forming acetaldehyde. So it leads to Wernicke-Korsakoff syndrome. Right? And this is again very, very important, that if a person is alcoholic and B1 deficient, immediately thiamine supplementation need to be given. So this is vitamin B1 TPP. Then moving on to the second vitamin, vitamin B2. Now again, vitamin B2 को क्या बोलते हैं? It is also known as riboflavin. Now, what is the active form? Again, we will come to that same format is there for every vitamin. The active forms of vitamin B2 are FAD and FMN. FAD stands for flavin, adenine, dinucleotide, and FMN stands for flavin, mononucleotide. Right? Now again, same pattern we are going to follow. Functions. If we look at the functions of B2, what are the biochemical functions? Okay? The most important, again, they are functioning as coenzymes. Now, this vitamin B2 is a coenzyme for so many important reactions. First of all, if we take FAD, it is required for as a coenzyme for succinate dehydrogenase. Right? Reaction क्या होती है इसमें? Reaction is that succinic acid is converted into fumaric acid, and FAD is converted into FADH2. This is a very important reaction, and this SDH enzyme, only enzyme of TCA, present in the inner mitochondrial membrane. Except this, all the enzymes of TCA, you are well aware of this, they are present in the matrix of mitochondria. And this enzyme has a very low redox potential. So this is one very important role of FA. Second important role is about FMN. Now, if we talk about FMN, this is the most important enzyme of oxidative deamination. किसकी? Of L-amino acids. And this is in fact the most important reaction in which FMN is acting as a coenzyme. And if you look at the reaction is this L-amino acid. Since it is auto-oxidizable, L-amino acid will be converted into an intermediate which dissociates to form alpha keto acid. Because this is a very important example of deamination, and this is auto-oxidizable. What do you mean by auto-oxidizable? On one hand, FMN is being converted into FMNH2. On the other hand, this is coupled with the degradation of hydrogen peroxide. And that is why this is known as auto-oxidizable. Right? So these are the two very important reactions in which riboflavin is acting as a coenzyme. Then third important reaction is one second. First second. And third is it is a component of ETC also. Component of ETC. Complex 2. I'll just write complex 2. So these are the important reactions in which FAD and FMN or riboflavin is used. Okay? Then coming over to the deficiency manifestations. There are 5 very important deficiency manifestations of riboflavin. First of all, there are 2 C's. One C stands for corneal vascularization. Right, there is a redness in the cornea. Second C stands for cheilosis. Cracks are there on the lips. Then cheilosis ke baad, these are 2 C's. Then dermatitis is there, seborrheic dermatitis. There is a rough scaly skin. Why? Due to desquamation. Right, 2 C's, then C, and then stomatitis is there. Fissures are there. In the mucocutaneous junction, and then glossitis is there. Glossitis is the color of the tongue turns to magenta or magenta colored tongue. Right, so we can say that 5 main deficiency manifestations are there: corneal vascularization, redness on the cornea, cheilosis, cracking of lips, right, seborrheic dermatitis, rough scaly skin, and then stomatitis is there. There are fissures at the mucocutaneous junction, and glossitis, the color of the tongue becomes magenta. So these 5 are basically, isko again I am telling you a mnemonic: 2 C's and then 2 S and G. So we can say that 2 C's, 2 S and G. CCSSG are the deficiency manifestations of riboflavin. So these are the 2 very important functions of B1 and B2. After this, the most important again feature is biochemical assessment. Of B1 and B2. Because how we have to estimate that B1 is not present in a particular subject or B2 is not present. तो कैसे identify करते हैं? There are different biochemical tests or we carry out the assay of the enzymes. सबसे पहले B1 के लिए obviously one is urinary excretion of thymine. If it increases, but the most important test which is carried out to check is it is erythrocyte ketolase activity. erythrocyte transketolase activity is reduced in B1 deficient subjects. Very, very important. I'll just highlight it also. Is reduced in B1 deficient subjects. This question has been asked in the exam. The B1 is such an important vitamin. So many questions have been asked on this B1. Right? And then coming over to B2. B2 में there is reduction in glutathione reductase activity. This is also again very, very important. Reduction in glutathione reductase activity. So if a question is asked like this, that biochemical test shows that it is showing a reduction in transketolase, the subject is B1 deficient. But if it is showing that there is reduction in glutathione reductase activity, the answer is B2. And these are the symptoms of B2, 2 C's, 2 S, and 1 G. Right? So this is about B1. Let me see if there is any other mnemonic related to B1 or B2. That's it. Clear? Then after discussing B1 and B2, then we come over to again a very, very important B complex vitamin, which is vitamin B3. Now, this vitamin B3 is actually most commonly referred to as niacin. And the most important feature about this niacin is it is a atypical vitamin. Now, what do you mean by the term atypical vitamin? Atypical vitamins are those vitamins which are synthesized in the body. There are only two vitamins which are typical, atypical vitamins. Examples are niacin and vitamin D. There are other vitamins also which are synthesized by the intestinal bacterial flora, but they do not come under atypical vitamins. जैसे you can say different, जैसे B12 is there, then intestinal flora में biotin is there, K is there, but they won't come under atypical vitamins. Atypical vitamins का simple सा example is B3 and vitamin D. अब if we talk about B3 or niacin, it is synthesized from tryptophan. Tryptophan, which is a essential amino acid. This, in the presence of enzyme QPRT, synthesizes niacin. And always remember 60 milligram of tryptophan is required to synthesize 1 milligram of niacin. This is again a very important question. This is the rate limiting enzyme. So we can say that niacin is synthesized from tryptophan. And 60 milligram of tryptophan synthesizes 1 milligram of niacin. And this is one of the very, very important atypical vitamin. Then now, after this, we will come over to what? Sources I am not mentioning unless and until there is a very important, or you can say different source. Just say vitamin B1 may I mentioned that the rice is the one which is the most important source. Otherwise, most of the eggs are there, then milk is there, then green leafy vegetables are there. They are all the common sources of all the vitamins. Then coming over to B3, Vesibi, it is an atypical vitamin. So we can say the diet which is having tryptophan will lead to the formation of niacin. Jaise, I'll just give you one example. There is, all of us are familiar with this maize or corn. Okay, so this maize has a protein called zein. Zein is the protein present in maize, and it is very interesting to know that it lacks tryptophan. So people whose staple diet is maize, who eat more of maize, they will be more prone to niacin deficiency. Kyun? Kyunki they are not generating tryptophan. Maize is having zein protein which lacks tryptophan. So if tryptophan is lacking, no niacin will be produced. Clear? So this is what is again. So we need to have those sources, and वैसे भी जो tryptophan है, this is a very important amino acid. It is an essential amino acid because along with the niacin, it also synthesizes serotonin. Serotonin, which is a very important vasoconstrictor, which helps to stimulate the cerebral activity. Similarly, melatonin, which helps in sleep and awake process, or it is a diurnal, a hormone which helps in diurnal rhythm. So these two are also synthesized from tryptophan. Right. Now, this is the sources. Then we come over to the biochemical functions. First of all, active forms. Yes, active forms. Now, if we look at the active forms of B3 or niacin, they are NAD, NADH, NADP, or NADPH. NAD stands for nicotinamide adenine dinucleotide. Same nicotinamide adenine dinucleotide, hydrogen is there, so hydrogen will come. Nicotinamide adenine dinucleotide phosphate, which has more energy obviously, and nicotinamide adenine dinucleotide phosphate along with hydrogen. So these are the 4 active forms of niacin. And if you look at the functions, now functions you can correlate so many functions. It's actually difficult to summarize the functions of niacin because it is the most important reductant which is present in our body. Most important. Most of the metabolism, they have to be dependent on NAD. If we talk about TCA, in TCA itself, there are 3 NADH reactions. And you can see 3, that means 3 into 2.5. 7.5 ATP are given by 1 TCA cycle. So you can see it is giving so much energy to the body. If we talk about lipogenesis or fat synthesis may be NADPH is required. In fact, whatever dehydrogenation reactions are there, they will always generate NADH or NADPH. So wherever you find pyruvate dehydrogenase, lactate dehydrogenase, simply write NADH or NADPH. So these are the biochemical functions. Now the questions which are mainly asked on niacin are the deficiency manifestations. Now, if we see the most important deficiency symptom of niacin is pellagra. Now, what happens in pellagra? It is characterized by 3Ds. And what are these 3Ds? Diarrhea, dermatitis, dementia, right? Diarrhea is simply loose stools will be there. Dermatitis, dry skin, scaly type of skin, and dementia is loss of memory. And these three can lead to the fourth D, which is death. Right? And again, the most important, this is a very important question which is asked in the exam. Basically, what is happening is there is hyperpigmentation wherever exposed surface will be hyperpigmented. And it forms a cassel necklace. I'll show you the image also. Cassel necklace is a characteristic feature of deficiency of niacin. You can see that. Diarrhea, dermatitis, dementia leading to death if untreated. And you can see that formation of cassel necklace is there. So this question has been asked in the exam that cassel necklace is due to the deficiency of which vitamin? So you can simply say it is niacin. One more picture is there. I'll just show you the picture. This is one more picture on which actually the question has been asked. This is the picture which is generally shown. So whenever this type of picture is shown, you can clearly say this is you can see the hyperpigmentation. This is the exposed area. And since it forms the shape of a necklace, so we say it is cassel necklace. So this is deficiency of niacin pellagra. Then after this, this is pellagra. अब one more thing is there, what are the factors which can lead to pellagra? अब देखो होता क्या है, again since it is synthesized from tryptophan, so we can say that if there is defective tubular reabsorption of tryptophan is there. If there is defective tubular reabsorption, we can say that urine of that patient will be having indole derivatives in the urine, and it will lead to Hartnupp's disease. So in Hartnupp's disease also, pellagra will be the main common symptom. It can lead to pellagra. Right? Then another important feature, carcinoid syndrome. Carcinoid syndrome can also lead to pellagra. Now, how carcinoid syndrome is related to pellagra? if we talk about the metabolism of tryptophan, we know that tryptophan metabolism is having two pathways. One is kynurin pathway and the other is serotonin pathway. We say maximum up to 3% of tryptophan is diverted to form serotonin, and kynurin pathway is required for the synthesis of niacin. Up, suppose if tryptophan is diverted to synthesize more of serotonin, what will happen? The concentration of serotonin will increase, and it will increase in the argintophene cells of GI, leading to carcinoid syndrome. Isn't it? Now, if excess of tryptophan is diverted to form the serotonin in the argintophene cells, the niacin synthesis will decrease. Again, if synthesis of niacin will decrease, it will lead to pellagra. So we can say that pellagra can be due to so many reasons. One is defective tubular reabsorption, defective synthesis of niacin, people whose staple diet is maize, and carcinoid syndrome will lead to pellagra. Right? ये तो हो गया the deficiency manifestations. Ab, this niacin being a atypical vitamin, it has a therapeutic role also. And what is the therapeutic role of this niacin? Therapeutic role of niacin. This niacin acts as a lipid modifying drug. Very, very important. Now, how it acts as a lipid modifying drug? It inhibits the action of lipoprotein lipase. That means if it decreases the activity of lipoprotein lipase, obviously what will happen? There will be decreased TGs. Right? So that is why it is a lipid modifying drug. Because lipoprotein lipase is the enzyme which is required for the degradation of lipoproteins. You know that lipoprotein which are degraded to form the fatty acids and glycerol. So obviously if this is inhibited, this will automatically decrease. Clear? So this is a very important role of niacin. And this is what is a atypical vitamin. And similarly, increased concentration of niacin is also harmful. If there is increased production of niacin, it is also harmful. Why? Because it increases the gastric irritation. It increases the acidity, leads to sometimes flushing also. Clear? So this is what is vitamin B3, niacin, atypical vitamin. The most important questions which have been asked on this are 60 milligram of tryptophan is required to synthesize 1 milligram of niacin. Then after B3, we come over to vitamin B5. Now, if we look at vitamin B5, this is also known as pantothenic acid. Pantothenic acid or pantotheniine. The other name is pantotheniine. Now, what is the most important point which we have to remember about this? That beta-alanine is a component of pantotheniine. In fact, it is a component of pantoic acid. Right. अब now again same active form तो हो गया. अब what is its active form? Its active form is SH-CoA. SH-CoA. This is the active form of pantothenic acid. Then coming over to the functions. Now, if we talk about the functions, all of us are familiar with acetyl-CoA, right? Then we are familiar with malonyl-CoA. We are familiar with propanol coenzyme A. And we are familiar with HMG-CoA. These are the 5 most important, you can say, derivatives of vitamin B5, and their function is acetylation. The function of this is acetylation, transfer of acetyl group. If we talk about acetyl coenzyme A, we know that acetyl coenzyme A is the intermediate of all the respiratory foodstuffs which we take in our diet. Whether we are having carbs, whether we are having proteins, or whether we are having fats, ultimately they are converted to form acetyl coenzyme A, which is utilized for the synthesis of fats again, which is utilized for the synthesis of ketone bodies, plus which is utilized for the synthesis of cholesterol. On the other hand, it undergoes degradation by TCA. So you can easily understand the value of acetyl coenzyme A, which is what? Which is derived from vitamin B5. So obviously, if you look at the deficiency manifestations, if B5 is absent from the diet, it will lead to lack of all the reactions which are related to the metabolism of fatty acids. Okay, then if we talk about malonyl-CoA, again we know that acetyl coenzyme A is converted into malonyl coenzyme A. Simple carboxylation is there, and the name of the enzyme is acetyl coenzyme A carboxylase. So I can say that if malonyl-CoA is not synthesized, no fat will be synthesized. Synthesis takes place. So B5 is important. If we talk about propanol coenzyme A, propanol coenzyme A is required for the synthesis of succinyl coenzyme A. And this succinyl coenzyme A is the precursor of heme. Again, we can say that if CoA is not there, this CoA is not there, heme की synthesis नहीं होगी. So you can imagine the importance. Right? This is the importance of vitamin B5. Then last is HMG-CoA. Very, very important. This HMG-CoA stands for beta hydroxy beta methyl gluteryl CoA. ठीक है? This is the most important intermediate of ketogenesis plus cholesterol biosynthesis, if you remember. And this HMG-CoA is produced when two molecules of acetyl coenzyme A combine with one more molecule of acetyl CoA. So it forms this HMG-CoA. And this HMG-CoA, if it undergoes splitting, that means the enzyme is HMG-CoA lyase, it forms the ketone bodies. And if this HMG-CoA undergoes a reduction, the enzyme is reductase and it forms mevalonate or cholesterol. So you can imagine again the importance of coenzyme A, because everywhere wherever CoA is written, CoA kahan se aa raha hai? It is coming from pantothenic acid. Right? And pantothenic acid is having beta alanine, because this question was asked in the exam that what are the components of pantothenic. So always remember beta alanine and pantoic acid. ये तो हो गए functions. Then we come over to the deficiency manifestations. Now, if we talk about the deficiency manifestations, the most important one is burning feet syndrome. It is also known as Gopalan burning feet syndrome. There is burning in the, feet. Just because of, again, you can say ki because these are the products which won't be produced without the absence of this B5. Right? So this is pantothenic acid B5. Then after B5 comes vitamin B6. Now B6 is also known as pyridoxine. But again, if we look at the active form of B6, the active form is known as PLP, which stands for pyridoxal phosphate. Because this pyridoxine or B6 exists in three forms. One is pyridoxine, pyridoxal, and pyridoxamine. When we put a phosphate here, this becomes the active form of pyridoxine, which is PLP. Now, again, after the active form comes the functions. Now, this PLP is a very, very important, you can say, vitamin which is directly linked to the metabolism of the amino acids. And we say that a rate of rate or RDA of protein intake is directly proportional to amount of PLP taken. Whatever RDA, whatever proteins we are having in our diet, they are directly proportional to PLP. That means PLP is connected with the proteins. Now, how it is connected, the question is that. So then we come over to the biochemical functions. Okay. Now see, the first and the foremost function of PLP is transamination. The enzyme of transamination is transaminases or aminotransferase. But the coenzyme is PLP. And the most important two reactions of transamination, one is alanine forms pyruvic acid, right? And glutamic acid. Here PLP is the coenzyme and aminotransferases is the enzyme, right? And the enzyme, if we have to classify this enzyme is alanine transaminase or ALT, or it is also known as serum glutamate pyruvate transaminase, which is a marker enzyme of liver diseases. So PLP acts as the major coenzyme. Then second reaction again of transamination is aspartate. देखो same reaction है, but the amino acid and the keto acid is different. This aspartate gets converted into oxaloacetate. Again, PLP is the coenzyme and aminotransferase, or you can say AST is the enzyme. Same alpha ketoglutarate and glutamic acid will be there. The enzyme is aspartate transaminase, or it is also known as serum glutamate oxaloacetate transaminase, which is a marker of heart diseases or cardiac marker. So this is one of the very, very important functions of PLP. Second important function of PLP is transsulfuration. What is transsulfuration? Let me just, I have underlined this, transsulfuration, which means transfer of sulfur group. Right? Now, what happens in this reaction? If we talk about transsulfuration, the most important one reaction is in the metabolism of methionine. If we remember, if we look at methionine, methionine is an essential amino acid. I'll just show you the reaction. What happens here is this methionine, which is a methyl donor, it is first of all activated to form S-adenosyl methionine. This S-adenosyl methionine then donates its methyl group and it is converted into S-adenosyl homocysteine. And this S-adenosyl homocysteine is then converted into homocysteine. Right? And this homocysteine is converted into methionine. This is a regeneration of methionine, which I have discussed in detail in the metabolism of methionine. Because here we are concentrating only on the vitamins. So here we are going to study what is the role of this B6 here. Okay. So now see what happens is homocysteine, it will combine with serine. Right? The enzyme here is cystathionine beta synthase. And the coenzyme here is B6. Clear? And it forms cystathionine. This cystathionine undergoes splitting to form cysteine and beta hydroxybutyrate. Now the question is, as you can see here, B6 is transferring sulfur. कहाँ से ट्रांसफर कर रहा है? होमोसिस्टीन में ये सीरीन ऐड कर रहा है एंड सिस्टीन एक्चुअली बीटा हाइड्रोक्सी ब्यूटाइरेट इज प्रोड्यूस्ड इन द एंड. 1 second. This is produced in the end. The major intermediate will be homoserine. Then it forms beta hydroxy butyrate in the end. Like this. So you can see the importance of B6. The reason why I am writing the individual reactions, because when we are going to study the deficiency manifestations, these are the reactions which will be hampered. So this is second important reaction, which is transsulfuration. Then third is metabolism of tryptophan. Now, if we look at the metabolism of tryptophan, just now I told you that tryptophan undergoes two different pathways. One is serotonin pathway, where it leads to the synthesis of serotonin. Second is kynurin pathway, where it leads to the synthesis of niacin. Because tryptophan is acting as both, means it is glucogenic as well as ketogenic. Now see, when we study the metabolism of tryptophan, kynurin pathway में 3-hydroxyl kynurin is converted into 3-hydroxyl anthranilate. Right? For this reaction, the enzyme is kynurinase. And the coenzyme for this enzyme is B6. I'll just highlight B6 here. So this is another very, very important reaction in which B6 is required. Then moving on to the fourth reaction, as a component of or you can say as a component of transsulfuration हो गया glycogen phosphorylase. And this is the only enzyme of carbohydrate metabolism which requires B6 as a coenzyme. मतलब ये जो B6 है, it is a component of glycogen phosphorylase. अब all of you know glycogen phosphorylase क्या है? It is a rate limiting enzyme of glycogenolysis. And rate limiting enzyme of glycogenesis, I'll just write it here because so that you again revise it. Glycogenesis is glycogen synthase. Okay. So this is again a very important, you can say, function of B6. Then the next function is B6 is a component of ALA synthase. Now, what is this ALA synthase? ALA synthase is the rate limiting enzyme of heme synthesis. And what happens is the reaction is this: glycine plus succinyl coenzyme A in the presence of enzyme ALA synthase and coenzyme B6 forms what? Aminolevulinic acid. Levulinic acid. So these are the 5 major reactions in which PLP is used as the most important coenzyme. Transamination, transsulfuration, you can say in the synthesis of ALA, then as a component of glycogen phosphorylase, and in the metabolism of tryptophan. And similarly, apart from this, it is also used as, I'll just write it. It's a component of decarboxylation also, for decarboxylation. Means removal of carbon dioxide. Ab dekho, most of the students get confused between decarboxylation and oxidative decarboxylation. Whenever in our body decarboxylation occurs, it is with the help of PLP. But whenever oxidative decarboxylation is mentioned, it has to be TPP. Let's say if I talk about decarboxylation, you can say this DOPA, dihydroxyphenylalanine, when it undergoes decarboxylation, it will form dopamine. Here, coenzyme will be PLP, not TPP. TPP कहाँ पे यूज होगा? TPP वहीं पे होगा जहाँ पे oxygen is also involved, means oxidative decarboxylation is there. Suppose if I talk about histidine, histidine in the presence of PLP will form histamine. This is again a kind of decarboxylation, but not oxidative decarboxylation. Then glutamic acid or glutamate in the presence of PLP will form GABA, which is gamma amino butyrate. Right? So these are some of the, and again you can say, matlab it's like, if you have to remember T square D, matlab T and D you can write. T square means transamination and transsulfuration. D stands for decarboxylation. And others are as a component of ALA synthase and as a component of, you can say, glycogen phosphorylase. ठीक है? अब ये तो हो गए functions. Now what are the deficiency manifestations? अब now see, if we look at the deficiency manifestations, I'll just take you to the reactions to make it more simple. Now see, coming over to the first reaction. अब देखो, if I say this PLP is absent, what will happen? No transamination will be there, isn't it? अगर transamination नहीं होगी, you know, transamination is the process which is actually related to the metabolism of amino acids as per their nitrogen skeleton, isn't it? Transamination leading to deamination, leading to the formation of urea. And that is what I told you in the starting, that protein intake is directly proportional to PLP. That means there won't be degradation of these proteins, isn't it? So there will be increased concentration of that. So first we can say that both the reactions will be suffered, and it will obviously lead to what? You can say this and this, again this won't be there. So there is no method of degradation of the amino acids. So it can lead to increased alkalosis also, because you know, if you look at the structure of the amino acids, they have both. They have a zwitterine, means they are amphoteric, they have more of amino group because there is no process of degradation of that. So increased concentration of amino group will be there. Okay? So one is we can write, if it go to the deficiency manifestation, first is we can write no degradation of the amino acids. ठीक है? Moving on to the second one. Second क्या था? Second was this. Now, if I say B6 is absent, what will happen? There will be increased concentration of homocysteine, and this leads to classic homocystinuria. So we can say that if B6 is deficient, classic homocystinuria is there, in which increased concentration of homocysteine is there, increased concentration of methionine is there. And this is detected by nitroprusside test. Clear? And you can say no cysteine will be produced also. Cysteine nahi banega, cystine nahi banega. Matlab you can see one thing is related to so many other things. And that is what is the importance of these vitamins. Then coming over to the next, I will again write it in the next one. Now, if we talk about the metabolism of tryptophan, what happens is, if B6 is absent, that is what we are concentrating on. That means there will be increased concentration of 3-hydroxylkynurenine. And this 3-hydroxylkynurenine will be diverted to form xanthurenic acid. So we can say that increased concentration of xanthurenic acid is an indication of B6 deficiency. Increased concentration of xanthurenic acid indicates B6 deficiency. Right? And in simplest language, if we say that a subject who is deficient of B6 will have xanthurenic acid in his urine, then next, this is so simple. You can say ki, since B6 is a component of glycogen phosphorylase, if B6 is not there, what will happen? Glycogenolysis will not be possible. If glycogenolysis will not be possible, glycogen will not be broken down. So it can also lead to hypoglycemia. Clear? Then component of ALA synthase. If we talk about component of ALA synthase, you can say if B6 is absent, I'll just write it. Actually, aminolevulinic acid is converted into heme. So if B6 is absent, heme synthesis will be defective. Defective heme synthesis. Right? Then if we talk about decarboxylation, decarboxylation may, if PLP is absent, there will be decreased synthesis of dopamine, and decreased synthesis of dopamine leads to Parkinson's. Right? If this reaction is not possible, histamine concentration decreases. We know histamine is secreted by the mast cells and it plays a very, very important role in hypersensitivity. And then if this reaction does not take place, GABA or gamma aminobutyrate, it is an inhibitory neurotransmitter. It will not be synthesized. Neurotransmitter, which can lead to convulsions or seizures, right? So you can see that what are the deficiency manifestations. So coming over to this, I'll just quickly summarize it. No degradation of amino acids, so you can say increased concentration of ammonia will be there. Second, we studied about, this was transamination, then yes, homocystinuria will be there. Then excretion of xanthuranic acid will be there. Uranic acid leading to xanthuranic aciduria. Leading to xanthuranic acid aciduria. Right? Then another is heme synthesis is impaired. Synthesis is affected. Right? Then after this, another convulsions or seizures will be there. Why? Because GABA is defected. Right? Then glycogenolysis may, being a component of glycogen phosphorylase, hypoglycemia will be there. So you can say that this one vitamin deficiency can lead to so many diseases, and that is what is the role of a coenzyme in the reaction. Clear? So this is vitamin B6, whose active form is PLP, pyridoxal phosphate. Now again I am telling you, don't get confused between decarboxylation and oxidative decarboxylation. Wherever oxidative term comes, means removal of carbon dioxide in the presence of oxygen, TPP is the coenzyme. But whenever we talk about plain carboxylation, PLP is used as a coenzyme. Then after B6, we come over to the next vitamin, vitamin B7. Now this is again a very, very interesting vitamin. If we look at the active form is simply biotin. Okay? As PLP is for transamination, biotin is for decarboxylation. Sorry, carboxylation. One second. जैसे PLP सबसे इम्पोर्टेंट कोएंजाइम है ट्रांसएमिनेशन का, वैसे ही बायोटिन इज द मोस्ट इम्पोर्टेंट कोएंजाइम for carboxylation, means one carbon up. And there is an antidote of biotin which is evident. Anti-egg injury factor. Actually, injury factor. This interferes with the action of biotin. So they say if a person consumes raw egg में this evidin is present, so it won't let biotin to function. So immediately if a person is having raw egg in the diet, immediately the supplementation of biotin has to be given, which reverses the action of evidin, because biotin is required for so many important carboxylation reactions in the body. ठीक है, अब एक्टिव फॉर्म अगर इसकी हम बात करें, इफ वी टॉक अबाउट द एक्टिव फॉर्म इज बायोसाइटिन, means it always carries that bicarbonate ions, because if we talk about carbon dioxide, it is present in the body in the form of bicarbonate ions. And you can say that, I'll just write biochemical functions. The most important function is carboxylation. Right? And for this carboxylation, always remember one mnemonic ABC. ABC. It stands for, A stands for ATP, B stands for biotin, and this C will stand for carbon dioxide or bicarbonate ions, like this. Means these three are the prerequisites of carboxylation. अब ये तो हो गया carboxylation. What are the important reactions of carboxylation? The most important enzyme or reaction is pyruvate carboxylase. Commonly, it is known as PC. If we look at the importance of this PC, we know that this is the only enzyme of gluconeogenesis which is present in the mitochondria. Mitochondria. ठीक है? This question has been asked in the exam. That is why I have mentioned it again. Only enzyme of gluconeogenesis present in the mitochondria. And all of you know what is gluconeogenesis. So I won't waste time explaining gluconeogenesis. We'll just concentrate on this reaction. So what happens in this reaction is this pyruvic acid is converted into oxaloacetic acid. 3 carbon atoms and it has 4 carbon atoms. One addition of carbon atom is there. And the enzyme is pyruvate carboxylase and the coenzyme is B7. And obviously ATP is required along with bicarbonate ions. So this is the entire assembly of conversion of pyruvic acid to oxaloacetic acid. And always remember, if B7 is absent, hypoglycemia will occur. This is very, very important because gluconeogenesis is the process of synthesis of glucose. Isn't it? So what happens is, जैसे अभी just now I mentioned a case कि if a person is having raw egg white and he complains that he's having some kind of breathlessness and all, immediately biotin supplementation is given along with, because the person can go into hypoglycemia. The reason is gluconeogenesis is hampered. And this is one of the very, very important anaplerotic reaction also, because whenever we are having a carb-rich diet, so what happens is excess of pyruvic acid will form oxaloacetic acid in reserve, so that when the body undergoes in a starvation state, some part of oxaloacetic acid is already there, which will undergo gluconeogenesis. So this is a very important reaction. Then second reaction is acetyl coenzyme A carboxylase. Again, one carbon up. सबसे पहले तो this reaction is a rate limiting enzyme. Yeah, this enzyme is rate limiting enzyme of lipogenesis. Very, very important. होता क्या है? एसिटाइल कोएंजाइम ए सिंपल बनाता है मेलोनाइल कोएंजाइम। ठीक है, 2 carbon atom, 3 carbon atom. Assembly required is enzyme is this along with what? B7. साथ में क्या बनता है? Obviously I told you the 3 components, bicarbonate ions and ATP. Because the question can be asked like this also, if the reaction is given to you and you have to fill in the blank what all are the coenzymes or the cofactors for this. So this is again a very important reaction. Clear? And the third carboxylation reaction which requires this is propenyl coenzyme A carboxylase. ठीक है, this is one of the most important reaction of gluconeogenesis, क्योंकि ये जो propenyl coenzyme A है ये कहाँ से आ रहा है? It is coming from the degradation of odd chain fatty acids. Odd chain fatty acids. Because whenever we are eating fats in our diet, we don't know whether we are eating even chain or odd chain. So whatever fatty acids are there, if they are even chain, they will be degraded to form acetyl-CoA. But if they are odd chain, they will be degraded to form propenyl coenzyme A. Now, what is the role of this propenyl coenzyme A? This propenyl coenzyme A will undergo again same process, carboxylation, enzyme is this, B7 is there and it forms methyl malonyl CoA. And this methyl malonyl CoA will then be converted into succinyl coenzyme A. We'll come to this reaction also because this reaction requires B12. We'll come to this reaction. But here we are considering about carboxylation. So this is carboxylation. So there are 3 important carboxylation reactions in our body. One is acetyl coenzyme A carboxylase, the rate limiting enzyme of lipogenesis. Second is propenyl coenzyme A carboxylase, which is again a very important enzyme required for the synthesis of succinyl-CoA. And third reaction is pyruvate carboxylase, which is the rate limiting enzyme, which is the most important mitochondrial enzyme of gluconeogenesis. Right? ये तो हो गया. This is what? This is simple carboxylation. Now what happens is there are certain biotin independent carboxylation reactions also in the body. जितने अभी तक हमने the 3 reactions which we studied, they require biotin. Without them, without biotin, carboxylation will not be there. But there are some reactions in which biotin is not required. So biotin independent carboxylation reactions. Because this question has been asked in your exam. biotin independent carboxylation. इसमें the first reaction is carboxylation of prothrombin. Very, very important. And this function is catalyzed by vitamin K. What happens is prothrombin mein glutamate is there, and this glutamate is carboxylated. This is done with vitamin K, not biotin. So this is one very important reaction which does not require biotin for attachment of carbon dioxide. Second is CPS1 and CPS2. These also does not require carbon dioxide. Now, if we talk about CPS1, CPS1 is an enzyme of urea cycle. Here what happens is, here ammonium ions plus bicarbonate, you see carboxylation is there, forming carbamoyl phosphate. Right? But ये जो bicarbonate है, biotin is not required. Here the enzyme is CPS1, carbamoyl phosphate synthetase 1. दूसरी तरफ, if you look at CPS2, CPS2 is an enzyme of pyrimidine biosynthesis. Again, in this also carboxylation hai, means bicarbonate ions are combining with glutamine to form carbamoyl phosphate. यहाँ पे enzyme क्या है? Here the enzyme is CPS2. Again, bicarbonate is there, but nothing to do with biotin. So these are the two important reactions. Then third important reaction is in purines, C4 and C5 of purines. Here also there is no use of biotin. ठीक है, यहाँ पे C4, C5 कहाँ से आते हैं? They come from glycine. Similarly, we can say C2 also, C6 also, it also comes from bicarbonate ions. But no biotin is required. ठीक है? So these are the biotin independent and those were the biotin dependent reactions. Ab, after this, we come over to the deficiency manifestations. Again, I have taken one picture. I'll just see biotin deficiency. See, the mnemonic is TRAIN. Okay, how you can learn it? See, TRAIN. T stands for topical fungal infection will be there. R, rash. A, alopecia will be there because B7 is required for the growth of the hair also. Then infectious susceptibility, we will be more susceptible towards infection, and nausea and vomiting will be there. These are the deficiency manifestations of biotin. And another very important feature about this is, ये तो mnemonic हो गया, then biotin deficiency also leads to ketosis. Basically, the person who is deficient of B6, deficient of B7 or biotin suffers from multiple carboxylase deficiency. Multiple carboxylase Deficiency. Very, very important. And the most important feature is alopecia, right? Bowel inflammation is also there. Then ketosis is also there. And the most important one is, it is also known as tomcat urine, because the odor of the urine changes. So we can say that tomcat urine is a characteristic odor in urine due to multiple carboxylase deficiency. And others you can see, train, you can remember it. Okay? Tropical fungal rash is there, then alopecia is there, because biotin is required for the growth of the hair, and then infectious susceptibility, and then nausea and vomiting. Baby B12, because there is inflammation in the bowel. Clear? So this is B7. Then one more thing is there, ki if this is again a very important point, ki that if a person is having a raw egg, right? So this raw egg will have avidin. This avidin is going to interfere with biotin. So if avidin is going to interfere with biotin, what will happen? That biotin will not function. And if biotin does not function, the most important reaction which will suffer will be this pyruvic acid to oxaloacetic acid. It won't take place. And if pyruvic acid to oxaloacetic acid does not take place, we can say no gluconeogenesis, which will lead to hypoglycemia. So you can see that how these are interrelated. So we can say that this hypoglycemia will be due to the inactivity of biotin. Why biotin is inactive? Because avidin is there, which is there to interfere with this. So even if a person is having raw egg, immediately biotin supplementation should be given. And if hypoglycemia occurs, automatically it will lead to this ketosis, and which will lead to this tomcat urine disorder. Right? So this is what is, you can say, biotin B7, which is the most essential coenzyme for carboxylation. So I'll just take you to this. These are the three important reactions which you have to learn by heart. That one is, you should know the mnemonic for carboxylation. Mnemonic for carboxylation is ABC. Means A stands for ATP, B stands for biotin, because carboxybiocytin, or it is the one which is actually transporting bicarbonate ions, and this stands for bicarbonate. Carboxylation के लिए there are three important carboxylation reactions. One is pyruvate carboxylase. You can say it is also a rate limiting enzyme of gluconeogenesis, or it is present in the mitochondria. You can see like this. Then acetyl coenzyme A carboxylase, very, very important rate limiting enzyme of fatty acid synthesis. You can see increase in the carbon atom, carboxylation. And then propenyl coenzyme A carboxylase, which converts propenyl coenzyme A to succinyl coenzyme A. वैसे भी this reaction plays a very important role. When we will discuss B12, I will tell you again the same reaction. because this is how the odd chain fatty acids are utilized by the body. Then I told you about biotin independent carboxylation reactions also. The most important one is carboxylation of prothrombin, as you can see. Then CPS1 and CPS2, very, very important. And then comes of purines, because purines may, you can say that de novo synthesis is there, that we have already discussed. And if we come over to the deficiency manifestations, you can say this is a mnemonic to remember the deficiency of biotin, and it can lead to ketosis, it can lead to multiple carboxylase deficiency. Alopecia will be there. This is a very, very important symptom of multiple carboxylase deficiency. Alopecia, bowel inflammation, and ketosis. And the urine starts giving the smell of that. So that is why it is known as tomcat urine disorder also. And then this is also very important. If a person is consuming raw egg, he has to be put on biotin supplementation. Otherwise, avidin will block the action of biotin. So biotin will not function, and wherever it is required, those reactions will suffer. So this is B7. So जैसे अगर if I have to summarize, I can say that जैसे B6 is for transamination, so as B6 is for transamination, B7 is for carboxylation. Remember this, for carboxylation. Now, so far we have covered B1, B2, B3, B5, B6, B7. Then we come over to hematopoietic vitamins, which come under again B complex. First is vitamin B9. Now see, this vitamin B9 is also known as folic acid, as its folic acid has been derived from the word folium. Folium means green leafy vegetables. Okay. Now again, what is the active form of folic acid? Always remember, active form of folic acid is THF and DHF. THF stands for tetrahydrofolate and DHF stands for dihydrofolate. Right? Now see again, what are the biochemical functions? Now always remember one thing, this folic acid plays a very, very important role in 1-carbon metabolism. Very important. Now, as the word suggests, 1-carbon means it is donating 1 carbon. So this becomes the most important biochemical function of folic acid. Now this 1 carbon can be given in the form of methyl group, which is CH3. It can be given in the form of formyl group, which is HCO. It can be in the form of methinyl group, which is CH2. Okay, because everywhere this is 1 carbon which is being donated. And that is why we say tetrahydrofolate whenever, and how we write it. Suppose if I write N5 N10 methyl THF, it means that it is carrying this methyl group. When it donates this methyl group, it will be converted into simple THF. So the methyl group has been given to the donor. So this is what is 1-carbon metabolism. And this is the most important function catalyzed by folic acid. Now, what are the examples of this 1-carbon metabolism? Now see, first reaction in this 1-carbon metabolism is formation of thymine from uracil. Because the first pyrimidine to be synthesized in the body is uracil. And the name of this we have already discussed in the metabolism of pyrimidines, that CPS2 and ATKs are the two rate-limiting enzymes which synthesizes uracil in the body. And this uracil is further converted into cytidine monophosphate, which is again a pyrimidine, and the most important thing is this uracil is converted into thymidine monophosphate, means UMP and TMP. So here if we look at the structure of uracil and thymine, simply there is a difference of methyl group. That means if we give methyl group to uracil, it will be converted into thymine, and that is what exactly happens. And this methyl group is being donated from folate. And the name of the enzyme here is thymidylate synthase. Thymidylate synthase. Very, very important. So we can say that this TMP is produced. And the most interesting fact, since we are discussing this reaction, most of the anti-cancerous drugs which are given to treat cancer are folate inhibitors, because they will check the multiplication of thymine in the cells, right? So anti-cancerous drugs are folate inhibitors. Right? So this is one of the very, very important reaction, which is an example of one carbon metabolism plus synthesized by folic acid. Then second important reaction is purine nucleus. If we talk about purine nucleus, we know that this second carbon atom and this eighth carbon atom, they also come from folate, and the group transferred here is formyl group or formate. That is why we say that folic acid is very, very important for the synthesis of the nucleotides, be it purines, be it pyrimidines. And that is why in deficiency of folic acid also, the synthesis of the nucleotides suffer. And that is why deficiency also leads to megaloblastic anemia. Right? So this is second reaction. Third reaction of folic acid is when glycine and serine have to be interconverted to one another. Here also the name of the enzyme is glycine synthase, but it requires folate. So this is also one of the very important reactions. And then fourth reaction is in the synthesis of methionine from homocysteine. This is a very important reaction because this is the only reaction which requires two vitamin B complex. One is B9 and the other is B12. So we will be discussing this reaction in B9 also, and we will be discussing the same reaction in B12, and you will see what is the main difference between the mode of action of B9 and B12. So then another is when homocysteine has to be converted, because if we look at the structure of homocysteine, it is without methyl group. Matlab, if we add a methyl group, homocysteine will be converted into methionine. So again, what is the donor of methyl group? Methyl group comes from THF. THF is folate. It will give this methyl group, and here B12 is acting as a coenzyme. means which is taking this methyl group, giving it to methionine synthase enzyme. To form methionine. So these are the 4 very, very important reactions in which folic acid is required. You can see conversion of homocysteine to methionine, conversion of glycine to serine in C2 and C8 of purine nucleus, and in the synthesis of thymine from uracil. Right. अब now see, ये तो हो गए. What are these? These are the functions. Then we come over to deficiency manifestations. Now here, if we look at the deficiency manifestations, we always say that folic acid is required, very much required at the time of pregnancy, because folate, again, because of this 1 carbon metabolism, is required for the development of the fetus. And if this function is not carried out, it can lead to neural tube defects. Spina bifida is there. Right? Then this is one very important aspect, and second is it can also lead to megaloblastic anemia. And the reason is due to a reduction in the synthesis of the nucleotides. So these are the 2 very, very important deficiency manifestations of folic acid. Right. Now, the most important topic again on which the questions have been asked on this folic acid is the biochemical assessment. The biochemical assessment of folate. Now, the most important test for detecting the deficiency of this folic acid is Figlu test. Right. And the other name of this Figlu test is histidine load test. These are one and the same thing, Figlu test and histidine load test. Now, what is this test? Figlu stands for formiminoglutamic acid. Right? And what happens is whenever this histidine undergoes breakdown, ठीक है, होता क्या है, this histidine, which is a very important amino acid in a sense that a slight change in the pH will be due to this histidine. It is the most stable amino acid. It's a basic amino acid and it has a characteristic imidazole ring in it. And this is the most stable amino acid at physiological pH. So this histidine, when it undergoes degradation, it forms urocanate. The name of the enzyme here is histidase. This urocanate forms an intermediate which is 4-imidazole 5-propionate. This intermediate forms this formiminoglutamic acid. Okay? Now just understand that this is a very simple reaction. This formimino group is given out. This is 1 carbon, and this reaction takes place in the presence of folic acid forming glutamate. Right. Now what is happening is, if there is absence of B9, folic acid is absent, there will be increased concentration of FIGLU, and this FIGLU will be excreted in the urine. This is FIGLU test to check the deficiency of folic acid, right? And that is why it is also known as histidine load test. Because what happens is, whenever we give a person, histidine orally to eat, so obviously if that person is deficient of B9, FIGLU will be excreted in the urine of that patient. Otherwise, glutamate will be produced, and you know that glutamate can be converted into alpha ketoglutarate. So histidine is a glucogenic amino acid. So you can see 2 questions can be asked on this particular thing, and this FIGLU is very, very important. So we can say that B9 deficiency leads to excretion of FIGLU in urine. That is why this test is known as FIGLU test or histidine load test. And this FIGLU stands for 4-miminoglutamic acid. And then second test for this biochemical assessment is ICAR, which is again a qualitative test. And rest is, we can make a smear also. So making a smear also, we can detect the presence of folic acid. Right? So these are the 3 very, very important tests to detect the presence of folic acid. And the function of folic acid is, it is responsible for 1-carbon metabolism in the body. And the most important reaction related to 1-carbon metabolism is synthesis of thymine from uracil. Right? So this is all about B9 or folate. Then we come over to another parallel vitamin, vitamin B. Now this B12 is also known as cobalamin. And if you look at its structure, it has 4 tetrapyrrole rings, corin rings we also call them, like this. So depending upon the central metal, it is named like methylcobalamin or cyanocobalamin or methylcobalamin, right? Now see what is the active form. As such, if we see, we cannot predict any active form to this vitamin because it is functioning like this. And this is the only B complex vitamin which is stored. Right. Now, how, and this is also absorbed, stored and absorbed. So how the absorption of this B12 takes place? What happens is, the parietal cells in the stomach releases a glycoprotein known as intrinsic factor. Intrinsic factor. And this intrinsic factor combines with B12. So basically it is helping in the absorption, and it is absorbed by the ileum. So maximum absorption, we can say, of B12 takes place in the ileum. An intrinsic factor is responsible for this, and this B12 is also known as extrinsic factor of Castle. This is very important. Most of the students get confused into this. This IF, intrinsic factor, is just required for the absorption of this, and B12, the other name of B12 is extrinsic factor of Castle, and this is how it is absorbed. Okay? Right? Now again, this is how we are studying it also. What are the biochemical functions of B12? So if we look at the biochemical functions, there are three functions or three important functions in which B12 is acting as a coenzyme. Now, what are these three reactions? First reaction is propanol coenzyme A to succinyl coenzyme. This is first very important reaction catalyzed by B12. Again, B12 का function क्या है? It will only act as a coenzyme, because starting से I am telling you, all the vitamin B complex which comes under B complex, they are functioning as coenzymes. Right? Now, if you look at this reaction, this is the reaction. This propanol coenzyme A, what is the source of this propanol coenzyme A? Odd chain fatty acids. Okay? This propanol coenzyme A undergoes carboxylation. Carboxylation I have already told you, just remember the mnemonic ABC, means B7 or biotin, and the enzyme is propanol coenzyme A carboxylase. I'll just write the name, propanol coenzyme A carboxylase. And B7 I'll write here. B7 is the coenzyme again. So here it will form dimethyl malonyl CoA. This dimethyl malonyl CoA, in the presence of enzyme racemase, forms L-methyl malonyl CoA. The enzyme is racemase. And the last reaction is, once this L-methyl malonyl CoA is produced, this in the presence of B12 and mutase forms succinyl coenzyme. Succinyl coenzyme A, which is forming heme. So you can say that the precursor of heme is succinyl coenzyme A, which is coming from propanol coenzyme A. Now here we are mainly focusing on B12. So this is one of the very important function of B12, that it helps in the conversion of propanol coenzyme A to succinyl coenzyme A. Moving on to the second function. This is required for, or you can say coenzyme for, ribonucleotide reductase. This is the second very important reaction catalyzed by this. Coenzyme for ribonucleotide reductase. Now this enzyme does what? It converts the NTPs into dNTPs. Means NTPs are the bases of RNA. It converts the bases of RNA to bases of DNA. Or you can say it converts a ribose sugar into deoxyribose sugar. Right? So this is again a very, very important coenzyme function of B12, that it is required for the conversion of NTPs into dNTPs. Then, because I told you it catalyzes only 3 reactions, so third reaction is conversion of homocysteine to methionine. This is a very important reaction, conversion of homocysteine to methionine. Now, exactly what happens in this reaction is this methionine, first of all, gets converted to its active form, which is SAM. This SAM donates the methyl group forming homocysteine, and this homocysteine needs to be converted into methionine. So this is a regeneration of methionine, क्योंकि methionine का सबसे important function क्या है? It is a methyl donor और it helps in transmethylation reactions. ठीक है, ट्रांसफर करता है मिथाइल ग्रुप because methionine is required for the synthesis of creatine. अब what role this B12 has to play in this? ये आ गया homocysteine, ये आ गया methionine. ठीक है, what happens is this methyl THF, which is folate, it is donating this methyl and it is being converted into tetrahydrofolate. Now what happens is this B12 picks this methyl group and then it donates it and it is converted into this. Means you can say that this methyl group, which is coming from folate, it is donating this methyl group or it is handing over this methyl group to B12 and itself it is being converted into tetrahydrofolate. B12 accept this methyl group and donate it to homocysteine in the presence of enzyme methionine synthase to form methionine, because homocysteine and methionine, if you look at the structure of homocysteine, if we add methyl group into it, it will be converted into methionine. So this is again the reaction catalyzed by B12. So you can see the, wherever I have highlighted, you can see the role of B12 like this. Now coming over to, ये तो हो गए इसके biochemical functions. Now what are the deficiency manifestations of this? Right? Now the most important deficiency manifestation is, we will take the first reaction first of all. First reaction में, if you see this, propanol coenzyme A is simply forming succinyl coenzyme A. If I say B12 is absent, if this B12 is absent, there will be increased concentration of methyl malonic acid, right? And this will be excreted in urine. So we can say that deficiency of B12 leads to methyl malonic aciduria. Very important. Deficiency leads to methylmalonic aciduria. Then deficiency of again B12 leads to pernicious anemia. Right? And third very important deficiency manifestation of this is non-classic homocystinuria. Homocystinuria. Now, whenever we study non-classic homocystinuria, we will just go to this reaction. If you see B12 is absent, like this, what will happen? There will be increased concentration of homocysteine, leading to homocystinuria. And if we see, the concentration of methionine will decrease. अब यहीं पे if we take one more reaction which I taught you in B6, I told you this was the reaction. Homocysteine in the presence of B6 forms cystathionine. And I told you that in B6 deficiency, again homocysteine was increased. But this was classic homocystinuria. ये क्या था? ये classic homocystinuria था. ये क्या है? ये non-classic है. And what is the difference? If you see both the images, you can jolly well correlate the differences. In classic homocystinuria, homocysteine increases, methionine also increases. Whereas in non-classic homocystinuria, homocysteine will increase, but methionine will decrease. So hypomethionemia will be there in a non-classic. In non-classic, methionine will be less, and in classic, methionine will be more. This is the major difference between classic and non-classic. Right? Ab here we are targeting only B12, so we will say that non-classic homocystinuria is there due to B12 deficiency. And this can also lead to folate trap. Very, very important. Now, what do you understand by the term folate trap? Because see, if you look at the reaction, okay, methyl group is being donated. Up, if B12 is absent, there is nobody to receive that methyl group. And folic acid is responsible for 1-carbon metabolism. So due to the trapment of folic acid, the cells start starving of folic acid. So we say that folate is trapped. Why folate is trapped? Because the carrier of that methyl group which is keeping folic acid intact or which is keeping folic acid active, it is absent. So, and this is the only reaction in our body which requires both. Means B9 and B12. And here you can say that there are so many, clinical cases or questions which can be formed on this. Methylmalonic aciduria will be totally due to the deficiency of B12. It has nothing to do with B9. And folate trap is the one which involves both B9 and B12. Clear? So we can say, how do we define folate trap? That methyl group is not free. Methyl group is not free because of B12 deficiency. So we say that, so tissues undergo starvation. Tissues are starved for folate. So no one carbon metabolism will be there, no methyl group will be donated, and it will lead to folate. Clear? So this is very important. And then again, if we talk about this, a very important feature is there, all the vegans or vegetarians, right? They are B12 deficient because the richest source of B12 is non-veg, meat basically. So vegans are more prone to B12 deficiency, right? So this is again one of the very, very important point about B12. And then ये तो हो गए reactions, deficiency manifestations. Then we come over to biochemical assessment of B12. A biochemical assessment में one is, we can say that abhi just now we have discussed that methyl malonic acid will be there. So if the subject is having methyl malonic aciduria, we say B12 is absent. Right? So we can say methyl malonic aciduria. Second is homocystinuria. For homocystinuria, the test is nitroprusside test. So it will be also positive. And second important test is the use of radioisotopes in this, and it is Schilling's test. In Schilling's test, we use cobalt 60. CO60. Again, we label it and we see whether it is absorbed or whether it is excreted in urine, depending upon the function of homocysteine. Right? So these two are the very, very important tests to check the presence of the level of cobalamin in the body. So here we finish up with all the B complex vitamins, in which we studied what? B1. If I have to make a chart, B1 whose active form is TPP, this is the active form, right? Functions are, if we study the functions, oxidative decarboxylation, and deficiency leads to beriberi. Right? Then we studied B2. B2 mein the active form was FMN and FAD. And functions are what? Electron donor or as a reductant. And if you look at the deficiency manifestations, I told you 2 C's were there, right? And 2 S were there. S and G. 2C means corneal vascularization, right? Cheilosis. 2S means seborrheic dermatitis, and then another S was, if you remember, seborrheic dermatitis, and G stands for glossitis, magenta color tongue. Clear? B3, niacin, NAD, NADP, a reductant. The most important deficiency disorder is pellagra. B5, active form is SH-CoA. Acetylation is the function. And B5 leads to burning feet syndrome. Burning feet syndrome. Comes B6, PLP. वैसे तो there are so many reactions which we have discussed, but I'll highlight the most important one, transamination. B6 का deficiency also we have studied with that chart. B7 biotin. Carboxylation we have again studied about carboxylation reactions which require B7 and carboxylation reactions which do not require B7. Alopecia and all or multiple carboxylase this thing is there due to B7 and it is also known as tomcat urine. Then B9 and B12. B9 is THF, B12 is cobalamin, and reactions we have studied because most of the reactions are common. Right? Now after this we come over to the last water soluble vitamin which is vitamin C. Very, very important vitamin. Now why it is important? Because first of all, what is the active form? It is ascorbic acid. Right? Now see, there is one very interesting feature about this, that vitamin C in humans, it cannot be synthesized. That is why it has to be provided from outside diet. But in non-primates, ascorbic acid is synthesized. Okay, I'll just write it in non-primates because this question has been asked in the exam. In non-primates, ascorbic acid is synthesized by uronic acid pathway. Uronic acid pathway, I have told you that this is the alternative pathway of oxidation of glucose. Okay? Humans cannot synthesize ascorbic acid. What is the reason behind this? Because uronic acid pathway is there in human beings, but uronic acid pathway stops to the formation of glucouronic acid, and that is why it is operative also in the body, because glucouronic acid is produced only by this pathway. And why humans cannot synthesize ascorbic acid? Due to the absence of L-gluconolactone oxidase. Due to the absence of L-gluconolactone oxidase. Okay, that is why ascorbic acid has to be provided from outside diet. Now the question arises, what are the functions? Why it is important? Now see, if we look at first of all the active forms, if we look at the active form of this ascorbic acid is actually being converted into dehydroascorbic acid like this. Dehydroascorbic acid. Right? So the most important function catalyzed by this is functions as an antioxidant. Right? Now we will see what are the biochemical functions or the reactions catalyzed by this ascorbic acid. Now see, biochemical functions. Right? Now see, the most important function catalyzed by this is hydroxylation of proline and lysine. The most important function. Now, what are these proline and lysine? They are the components of collagen. Components of collagen. We all know that collagen is the most abundant connective tissue which is synthesized in the body, and it has a triple helical structure. And if you look at the amino acids present in collagen, it has repeats of glycine, proline and lysine. Right? And collagen is a required, since it is a connective tissue, it is required for the, it gives, a kind of firmness to the teeth also. The tooth is embedded in a socket which has collagen. So it gives strength to the tooth also. Okay. So now collagen is a protein, so it is synthesized in the body. Now, we have already discussed that there are some kind of post-translational modifications also. Post-translational modification means the protein which is produced, it has to modify itself before it has to undergo some function. So, same is the case here. So, what happens is this proline is converted into hydroxyproline or hydroxyl proline. The entire strength of collagen is determined by measurement of this hydroxyl proline, which is the question which has been asked in the exam. And lysine is converted into hydroxyl lysine. Obviously, the enzyme would be what? Here it will be prolyl hydroxylase. Here it will be lysyl hydroxylase, right? But the most important component of these two enzymes, one is vitamin C along with iron, along with molecular oxygen. So these three are the components of hydroxylation. So vitamin C plays a very, very important, you can say, function in hydroxylating or hydroxylation. Then coming over to the second function. Second function is it helps in the action of this enzyme also, tryptophan hydroxylase. Now this tryptophan hydroxylase is required for the synthesis of serotonin and melatonin. So again, vitamin C is there. Right? Then third is tyrosine hydroxylase. Hydroxylase. Again, this is required for the synthesis of catecholamines. Again, vitamin C. Right? Then, what is the other function? It helps in absorption of iron. Basically, it helps in the conversion of ferric to ferrous. This is also again vitamin C. So this is also its function. Right? Then another important function of this is it prevent hemolysis by converting meth HB to HB. Again, vitamin C. ठीक है, and in a nutshell, always remember one thing, wherever hydroxylation is there, जैसे right now तो I am telling you about catecholamines, norepinephrine and epinephrine. Similarly, in the synthesis of serotonin and melatonin, wherever we write hydroxylation, the most important coenzyme is vitamin C. ठीक है, so then it prevents hemolysis also. So you can see that there are so many important functions. मतलब I can say in collagen synthesis, in catecholamine synthesis, in absorption of iron, in formation of melatonin, in formation of serotonin. And it converts met HP to HP. So it is preventing the hemolysis also. So these are the functions. Now after the functions, we come over to what? The deficiency manifestations. Now see, first of all, if we talk about collagen synthesis, it leads to scurvy. What happens in scurvy is the tooth becomes, there is, swollen gums are there, right? They start bleeding also. Now, what happens is, why they start bleeding? Because collagen formation is defective there. Collagen formation is defective. Leading to that sponginess. The gums becomes spongy, swollen, they start bleeding. Because that collagen formation is defective, then the tooth cannot remain in that socket, so it starts moving. Then this also leads to petechiae also, because there is again formation of small granules because of that excessive hemolysis is there. Then delay in wound healing also is there. Delay in wound healing. Again, reason is collagen formation is defective, right? Delay in wound healing and increase in the bleeding time is there, right? So these are some of the deficiency manifestations. Then one more I told you about, that since it increases function care, it increases the absorption of iron. So deficiency of this will lead to microcytic anemia. Right. And then one more is there. There is one more type of scurvy which is known as infantile scurvy. Now, what happens in this infantile scurvy? Okay. The main difference between the scurvy in adults and infantile scurvy is no gums bleeding. Reason क्या है? Because teeth are not erupted. So what are the other symptoms of the infant who is suffering from vitamin C deficiency? Pallor will be there, right? Diarrhea will be there. And skeletal deformity will be there. Skeletal deformity. Right, so these are the deficiency manifestations which are caused by the deficiency of vitamin C. Now see, here one more question is asked about what is the richest source of vitamin C. So always remember, generally all citrus fruits citrus fruits are rich in vitamin C, and amla is the richest source of vitamin C. Up sometimes what happens is, if the concentration of vitamin C increases, or you can say toxicity of vitamin C, or this is also known as hypervitaminosis, means generally in all the B complex vitamins we didn't study about this toxicity because they are, once their function is over, they are excreted also in urine. But this vitamin C plays a very important role in hypervitaminosis, because if we increase the RDA of this, so what happens is this ascorbic acid is converted to dehydroascorbic acid, and this dehydroascorbic acid is converted into, degraded to form oxalic acid. Okay, so if the concentration of ascorbic acid increases, this increases, oxalic acid increases, and it leads to the formation of oxalates. So oxalate formation is one of the very, very important features of increased intake of vitamin C. Then second is, because it helps in the absorption of iron, though the second is iron overload will be there. This will be also one of the important features of vitamin, right? So obviously that is why it is recommended to have vitamin C in the recommended dosage only, and it can finally lead to gastric irritation or flatulence will be there. Hyperacidity will be there due to increased intake of this. Right? So this is vitamin C, which plays a very, very important role in the synthesis of important compounds, and how it plays an important role by catalyzing the hydroxylation. Right? So let's quickly see what have we studied in this. See, first of all, vitamin C, ascorbic acid. I'll just see if some image is there, so that it will be easy. Yes, one image is there. We'll discuss this. Because if you try to understand the mnemonics, it becomes easy. See, 4H, deficiency of vitamin C, hemorrhage, hyperkeratosis, hypochondriasis, and hematological abnormalities. And mainly the reason is due to defective collagen formation, because it is required for the hydroxylation of proline and lysine. And this question has been asked in the exam also, that proline and lysine may hydroxylation requires vitamin C, and along with vitamin C, what are the other components? So the other components are molecular oxygen and iron. And vitamin C always increases the absorption of iron. That is why vitamin C had a very important role to play during COVID also. Right? So let's see. But human beings have to have vitamin C in their diet because of the absence of L-gulonolactone oxidase. This question was also asked in the exam. Okay, and this is the active form, ascorbic acid to dehydroascorbic acid, and vice versa. And that is why it functions as an antioxidant. And the functions are hydroxylation of proline and lysine as a component of tryptophan hydroxylase, as a component of tyrosine hydroxylase, helps in absorption of iron, and it prevents hemolysis by converting methemoglobin to hemoglobin. And that is why the deficiency manifestations, mainly it is scurvy, and infantile scurvy is also there. Scurvy may obviously swollen gums or bleeding will be there in the gums because collagen formation will be defective and it will be like spongy, PTSD will be there, delay in wound healing. That is why there will be an increase in the bleeding time. And why it is, since it is related to the conversion of ferric to ferrous, it will lead to microcytic anemia, microcytic hypochromic anemia also. And infantile scurvy, if we see, no bleeding in the gums because teeth are not erupted at that age. Pallor will be there, diarrhea will be there, and skeletal deformities will be there because again there will be defective formation. And richest source is this. And toxicity of vitamin C is also very important because overload of iron will be there and there will be formation of oxalates. Hyperacidity will be there and it can also lead to flatulence. Gastric irritation will be there. So here we finish up with the water soluble vitamins. And all the water soluble vitamins are functioning as coenzymes.